Hi, welcome to part 2 of this 3-part introduction to the physiological principles module. Please make sure that you've watched the first part and taken any notes before you start this part.
The human body is made up of carbon-based molecules. These are the so-called organic compounds. Carbon binds to other elements to form a very diverse range of molecules, some of which can be large macromolecules. These molecules form different families of compounds according to common chemical properties. Carbohydrates include sugars and starches. Lipids are an essential component of many structures and chemicals. Amino acids are the small subunits that form chains that go on to form proteins. Nucleic acids formed the DNA and RNA. The name carbohydrate relates to their composition. So you have carbo for the carbon, hydrate for the water component. Carbohydrates form only about 3% of the body's organic matter, but they play a really central role in the chemical reactions that provide cells with energy. They're classified according to the size of their molecule. They include things like sugars, glycogen, starch, and obviously not in humans, cellulose. Sugars are typically monosaccharides or disaccharides. So you can have single saccharides, this is a single unit, if you like, of carbohydrate, or large collections of saccharides bound together to give you glycogen, starch, and in plant cellulose, which are polysaccharides. Oligosaccharides are sugars bound to other molecules, so other components, such as glycolipids. In this case, glyco refers to the carbohydrate, and lipid, of course, is fat. You can also get glycoproteins or even glycolipoproteins. Monosaccharides are simple sugars. They have 3 or 6 carbon atoms. So a good example is glucose. Glucose is a 6-carbon monosaccharide. It's known as a hexose, hex for 6. The chemical formula of glucose is C6, so 6 carbons, H12O6. So we've got 12 hydrogens and 6 oxygens. Remember that carbohydrate is carbon and water. And the chemical formula of water, of course, is H2O, i.e. twice as many hydrogens as there are oxygen. So hence the H12O6. Have a look at the diagram. See if you can pick out the 6 carbons, 12 hydrogens and 6 oxygens. Fructose is also a monosaccharide. Enzymes can remove water from monosaccharides. When that happens, you get what's called a condensation reaction, and 2 monosaccharides can be bound together. This can make a disaccharide. An example of this is milk. Milk is a lactose. It's made up of 2 monosaccharides, glucose and galactose, bound together. Polysaccharides, this is where we get lots of monosaccharides, and they're all bound together in a long chain. This makes them very large. It makes them macromolecules, and they're often used for storage. And a classic example is glycogen. Glycogen is made up of glucose. It's lots of monosaccharides bound together into these branching chains. It's then stored away in the liver. And what this means is that between meals, like Milky Way, you can use it to give you energy between meals and to prevent hypoglycemia between meals. Another example of a polysaccharide are the starches. So starches like the flour in your bread. You break that down once you ingest it into monosaccharides. This gives you energy. So let's look at carbohydrates on the whiteboard.
Carbohydrates then. Carbohydrates, as we've seen, are really just sugars. And we've talked about carbohydrates being carbon and water, hence the name. Carbon and water equals carbohydrate. Yeah? Carbohydrates made of carbon and water. So our simplest form is our monosaccharide. So monosaccharides have 3 to 6 carbons only. And examples that we can have of this are glucose with 6, or with 6, fructose. Under certain circumstances, glucose can become fructose within cells. So these are simple monosaccharides. Monosaccharides. And basically, they look like this. They're that straightforward. Except when I draw them, in which case they don't look very good. But you get the idea. Yeah? Monosaccharides. Our next most complex, I suppose, or complex is a bit of a joke, are disaccharides. Disaccharides. Again, pretty straightforward. We have 2 monosaccharides joined together. Yeah? Well, I'm running out of sides. There we go. And now another one. They're joined together through that condensation reaction. So, in other words, we're talking about the removal of water. And we join them together. And of course, the example we talked about was lactose, which is glucose and galactose. I'm glad I made that one slightly bigger. I can fit galactose in. So these are our disaccharides. And of course, that takes us on to the really, really big ones. We're talking about polysaccharides. Polysaccharides, this is where we start to come out of simple sugars. And we're talking about starch. We're talking about glycogen. We're talking about things that can store little saccharide units for use later. We can break them down. Yeah, so we have our polysaccharides. And our key example, and it's a really, biologically, it's a really important example, is glycogen. Glycogen, we take glucose, we stick all the glucoses together, we store it in the liver, and it's the sweet you can eat between meals. It's our store for when, okay, it's a few hours until I eat. I need some glucose, I'll break down the glycogen. And these polysaccharides form generally great big branching chains. So we could go on for some way here. Yeah. Each of those is a little saccharide. Each of those we can basically come along and we can chop those off. And that releases us glucose. So these are really storage molecules, and it tends to be the starches and glycogen. The only other one we did talk about was oligosaccharide. Right. And this is basically anything that is a sugar with something bound to it. Yes? So we talked about glyco. ... So we can have glycolipid, we can have glycoprotein, we can have glycolipoproteins. So this is where we have a sugar and we bind something to it. And quite a few of them are very important structures in terms of the body.
Lipids. So lipids, we have triglycerides. These are fats. Oils. Oils are liquid fats. Waxes. An example of that is possibly earwax or cerumen. And phospholipids. Phospholipids form the plasma membranes and the membranes within cells. Steroids. Here we're talking about potentially hormones. Generally, their functions include providing a source of energy. Fats are a very good source of energy. Signaling and forming, as we said, like the phospholipids, the plasma membrane of cells. Lipids overall are hydrophobic. They don't like water, or they are insoluble in water. Think about when you put a greasy pan into water, or the lipid jumps off and floats on the surface. But generally they may have a hydrophilic, so they like a portion that likes water, region that can interact with water. Anything that is hydrophobic will be lipophilic, so it won't like water, but it will like lipid. So first on our list were triglycerides. Triglycerides have two components. We have a 3-carbon glycerol molecule. This is the blue vertical line on the diagram on the left, attached to 3 fatty acid chains, hence the term triglyceride. They're transported around the body in little casings of phospholipid and lipoprotein, and these little casings are called LDL, low density lipoprotein. The density is all about the proportion of proteins to lipids. So very low density lipoprotein has fewer proteins but more lipid, and this is what LDL starts its life as when it's released from the liver. Generally, triglycerides are stored within fat cells, adipose cells, as lipid droplets. They can then be broken down for energy. Both glycerol and the fatty acids can be used to provide energy for cellular functions. In fact, they're better than glucose. The process of breaking them down is known as lipolysis, and there are various enzymes within adipose cells that are involved in this process, but one of the main ones is a lipase called lipoprotein lipase, and this separates the glycerol from the fatty acids. Phospholipids are similar really in structure to triglycerides. You have a glycerol molecule at the top, but you also have a phosphate group, hence the word phospholipid. Instead of 3 fatty acid chains though, you have 2, and these are hydrophobic. They don't like water. Phospholipids form the phospholipid bilayer of cell membranes, the plasma membrane around cells. It also forms all the membranes within cells, so that all of those membranes can interact because they're exactly the same. Because the fatty acid chains on the inside are hydrophobic, and the glycerol and phosphate group on the outside are hydrophilic, the membrane of cells forms a closed envelope because the lipid tails are trying to keep away from the water, whereas the phosphate groups and the glycerol are quite happy in contact with the water within the cell and the water outside the cell. Steroids are also lipids. They're very different in structure. Instead of the heads and tails, we have 3 hexagons and 1 pentagon. And all of these are carbon atom rings. Of these, atoms can be bound, and that determines the type of steroid. Many steroids are hormones. For example, cortisol, which is a glucocorticoid, aldosterone, which is known as a mineralocorticoid, and also the precursors of the sex hormones. And all of these are produced by the adrenal cortex. Bile acids are the major component of bile, and these are also steroids. Cholesterol is a particular subgroup of steroids, known as sterols. Cholesterol is the precursor of many other steroids, but it can also be synthesized when we need it, if it's low. It forms rafts within the plasma membrane. So little rafts of cholesterol sit within the lipid tails of the plasma membrane to stabilize it. And it's also transported around the blood within LDL.
So let's talk about lipids then. Lipids, we have, as we said, we have our triglycerides. Triglyceride. And this is our simple fats. Triglyceride, the 3 is a bit of a giveaway because we've got 3 fatty acid chains. We've got a 3 carbon glycerol molecule at the top. So all those would be, let's put the little C's in, carbon, and this is our glycerol. Below that, we have fatty acid chains. And all of these can be separated, and both of these, both the glycerol and the fatty acid chains, provide energy during cellular respiration. And actually, they're a lot better at it than glucose. There we go. So we have our fatty acid chains. There you go. Triglyceride, 3 fatty acid chains. Through the process of lipolysis, we can separate them. But we'll talk about that during the module. So something that looks very similar is our phospholipids. So phospholipids, Whereas these are going to provide us with energy. Phospholipids, well, they can provide us with chemicals, but generally we think of them as providing structure. They are the structure of the plasma membranes in the cells. So phospholipids actually look very similar. So let's have our phospholipids. So again, we have a molecule of glycerol. Generally, all fats have glycerol. So we'll have our glycerol. But phospho, as the word might suggest, we also have a phosphate. Phosphate is basically we have one phosphate and we have loads of oxygen molecules. So it's called a phosphate group. So we have glycerol, there you go, borrow that arrow. And in this case, whereas here we had 3 fatty acid chains, we only have 2 in terms of phospholipids. So let's have one there and one there. 2 lipid tails. And these form the center of the plasma membrane. The glycerol and the phosphate are on the outside, either side of the plasma membrane. We'll talk about that in the next part. Okay, so the one that we haven't talked about, and don't I just love drawing this, are steroids. So steroids, basically we have different types. Most of them are hormones, but also bile acids, cholesterol. Cholesterol is also a steroid. And we have, instead of these chains, we have carbon rings. We have 4 carbon rings. So we have one there, and we might have one there, and we'll have another one there. Yes, so that's our 3, and then we might add on, we actually have a little other one there, and this is more of a pentagon, excuse the drawing. Yeah, so we have, that's the sort of, if you like, the basic. Then we have various bits sticking out, and these bits sticking out have other molecules attached to them which determine the type of steroid it is. Most of these things will contain carbon, oxygen, and hydrogen. Hydrogen. In different configurations that give us a different type of steroid. So we have some very different shapes there, but all of these are our lipids.
So there are 21 amino acids. Amino acids, most of them come from the diet, but we can synthesize them within the body when necessary. Only 9 can't be synthesized. These are known as essential amino acids because they have to come from the diet. Amino acids have atoms of hydrogen, carbon, nitrogen, and oxygen. The R group determines the specific amino acid. Look at the picture to see the R group. The rest is the core. That stays the same in all amino acids. The R group can be transferred, and this is done by liver enzymes, and this will create different amino acids. The process is called transamination. Amino acids can be used like lipids and like glucose to make energy. Or they can be a source of carbon or nitrogen atoms for other biochemicals. Most of all, they form complex proteins, and they're bound together during a process known as protein synthesis that we'll talk about within the module. They're bound together by peptide bonds. This is amino group to carboxyl group on the next amino acid. This creates either small peptides of a few amino acids or long and branching chains of amino acids known as polypeptide chains. Polypeptide chains can then be folded into an increasing complexity of shapes to become helices, pleated sheets, or in a more complex form, they can become tertiary structures or combined with other polypeptide chains to become highly complex quaternary structures. They can be structural or chemical. So structural proteins may form channels in the plasma membrane, tubules within the cell, receptors on the plasma membrane of cells, lots of different structures. Chemical proteins could be hormones, neurotransmitters, enzymes, lots of different chemicals. Nucleic acids are DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. They are macromolecules, and both are chains of nucleotides. Nucleotides have three components: a pentose sugar, so a 5-carbon atom sugar. This is known as a ribose. In DNA, the ribose lacks an oxygen atom, hence deoxyribonucleic acid. They also contain a phosphate group and a nitrogenous nucleobase, and this is specific to the nucleotide's base type. Look at the diagram and identify the three parts. The nitrogenous base is the nucleobase. It can make the nucleotide adenine, cytosine, guanine, or thymine in DNA. In RNA, thymine is replaced by uracil. The sequence of the nucleotides in the chain that forms DNA or RNA is known as the genome. It provides the genetic information for protein synthesis. And during cell replication, mitosis or meiosis, DNA is replicated to produce gametes. The pairing of bases in the double helix strand of DNA, plus between DNA and RNA, and then RNA to RNA during protein synthesis, was determined by Watson and Crick in 1953. See if you can work out by the colours what is pairing to what in the DNA double helix in the diagram. Yes, Cytosine pairs to guanine, adenine pairs to thymine, or uracil in RNA. An easy way to remember this is C is a curly letter, as is G, so C to G. A and T are spiky letters, so A to T. The DNA helix is usually found in the nucleus of cells as spaghetti-like chromatin. Generally, chromosomes only form just before mitosis or meiosis. DNA is also found in a circular form in the mitochondria within cells. Mitochondria are important organelles that are often called the powerhouses of the cell. The helix forms a circle so that the mitochondria can replicate to make more energy for the cell. Mitochondrial DNA is maternal DNA, so more of our genome actually comes from our mother, and this is known as Eve theory. RNA comes in three functional structures. Messenger RNA, or mRNA, pairs to DNA in the nucleus to form a template for protein synthesis. The nucleotides of RNA pair to the nucleotides of DNA, C to G and A to T or U. Ribosomal RNA forms the ribosomes that read messenger RNA, and the ribosomes are found outside the nucleus. Transfer RNA delivers amino acids in the correct sequence. Each transfer RNA carries a specific amino acid that is bound to other amino acids to form a polypeptide chain. This polypeptide chain is then used to make the complex proteins we were talking about before. A series of 3 nucleotides are known as a codon in both RNA and DNA. The nucleotides can be a combination of any of the nucleobases. Each codon then codes for a specific amino acid. Have a look at the diagram for a minute. The line of RNA at the top could be messenger RNA, the template taken from DNA. The sequence of nucleobases, G, C, U, A, C, etc., is divided into codons of 3 bases. So codon 1 is guanine, cytosine, and uracil. Uracil rather than thymine because this is RNA. This codon, GCU, is specific to the amino acid alanine. Codon 2, formed of adenine, cytosine, and guanine, ACG, is specific for the amino acid threonine. The codons code for those specific amino acids. This section of RNA, therefore, would create, or code for, a polypeptide chain, i.e. a chain of amino acids, made up of the amino acids, reading from the left, alanine, threonine, glutamate, leucine, arginine, and serine. We will talk about protein synthesis in more detail at the start of the module.
So, acids, amino acids and nucleic acids. Amino acids are what build our proteins. They're the little units that bind together in great long chains. Those great long chains get folded into complex shapes to become either chemicals or structures. But the basic unit is the amino acid. So we have a hydrogen atom, we have a carbon atom, we have on one side something called a carboxyl group. In case you're interested, minus COOH, that's the chemical equation for its molecule, its group. On the other side, we have an amino group. The amino group contains nitrogen, it's nitrogenous. So it's NH2. Okay, so that's the boring basic amino acid. There you go. Let's get a bit of interest into our amino acid. Joined to the carbon, bound to the carbon, we have something called an R group. It's the R group that determines what specific amino acid it is. So there are different R groups for all of the different 20 amino acids. So it might be something like alanine, serine, etc., glycine. Any of them you'd care to mention, this R group is what it makes it specific, specifically makes that amino acid. The interesting thing is, enzymes in the liver, through a process called transamination, can take the R group from one base and move it to another one. So they can change amino acids from being one type to another type. And it's a key function in the liver. And you all keep testing liver enzymes. Those are the sort of enzymes that you're testing for. Okay, so that's our amino acid. They make proteins. Join them together with peptide bonds. Carboxyl will join to an amino group, and then so on and so on in a great long chain. Yeah? Which brings us to nucleic acids. So nucleic acids form, yes, DNA and RNA. DNA, RNA. The only difference, what makes the difference, is the type of sugar in this molecule. So we will have a phosphate group. We've already met phosphates over here, and then that is then bound to a sugar. This is always a pentose sugar, so a five-sided sugar. 1, 2, 3, 4, 5. Five-sided sugar. That's a pentose. Now, what determines the DNA or the RNA is whether this is a ribose RNA, ribonucleic acid, or a deoxyribose, deoxyribonucleic acid. Yeah? Okay. Let's get a slightly different colour for this bit. Now, this is the bit a bit like the amino acid that determines what it is. So attached to that, we have a nitrogenous base. In other words, it's got nitrogen in it. This nitrogenous base can also be called a nucleobase, because the whole thing is making up, this whole thing is a nucleotide. Nucleotide. What determines what type of nucleotide it is the nuclear base, the nitrogenous base. Nucleobase. Okay, we don't have 20 nucleobases. We have, well, actually, we have 5. So, we have adenine, thymine, thymine in DNA, in RNA, that's uracil. And then we have cytosine and guanine. So 4, 5, depending whether it's DNA or RNA. Yes? Thymine in DNA, uracil in RNA. Now these will pair in specific ways. So our A, adenine, only pairs to thymine or uracil. Cytosine only pairs to guanine. Easy way to remember this. Spiky letter binds to spiky letter. Curly letter binds to curly letter. Just easy to remember that way. So these only pair in this way. And of course, what we're talking about pairing here is in some cases DNA to DNA. So here we're talking about the helix in the nucleus of DNA. The helix is paired in this way. Also in the nucleus, we might get DNA paired to RNA, because what we're doing is we're making a copy of the DNA out of messenger RNA. So in terms of RNA, we have messenger RNA, mRNA. We have our ribosomal RNA, which forms the ribosomes. That's not a very good R, is it? There we go. And we have transfer RNA. And transfer RNA is what brings in the amino acids. Once we get out to the ribosomes, transfer RNA codes for different amino acids. That coding is dependent upon the sequence of nucleotides. So say we've got a sequence of 3. A sequence of 3 is always called a codon. That 3 might be, I don't know, adenine, cytosine, and guanine. ACG. That will code for a specific amino acid. So we have codons of 3 nucleotides Now, those nucleotides might all be adenine. That's fine. But any combination, there's 60-odd combinations of these nucleotides. So codons of 3 nucleotides code for, or are specific for, 1 amino acid. So they code for a specific amino acid. And this is where we get our link between the two of them. Yes? So nucleic acids code for amino acids. They tell us what proteins we're going to make.